mainly focus on oogenesis and also on the procedures before the fertilization. So actually, oogenesis, in the name itself, you can see it is formation of female germ cell or female pronucleus. So actually, this oogenesis begins at like in intrauterine life. So even in the male, if you remember, it is beginning at intrauterine life at 8th week, and it is getting more in 15th week. It is elevated in 15th week in male, but in the female also it will start in the intrauterine life. The first one, it's a oogonia. This oogonia will first undergo the process of mitosis. So after the mitosis, this oogonia will convert into primary oocyte. Okay, so this primary oocyte now will undergo the process of meiosis 1. So first it has undergone mitosis. So actually in the meiosis 1, what happens in the case of female is this meiosis 1 is struck in the prophase 1 of diplotin stage. Prophase 1 diplotin. So actually in this stage itself it is stopped. So till puberty, like everything is stopped, like here only in intrauterine life it has come till meiosis 1 and it is stopped in diplotin prophase 1. So actually at the puberty, what happens is in the puberty time there is activation of hypothalamus axis. When hypothalamus is activated, it will produce gonadotropin releasing hormone. It will act on anterior pituitary and it will lead to release of both FSH and LH. Especially LH hormone will lead to surge of LH hormone, luteinizing hormone surge will mainly lead to the completion of meiosis 1 process, and as the meiosis 1 process will complete, the further it will form into secondary oocyte. Along with secondary oocyte, there is also formation of our polar body. This is important. So mainly because of LS surge, what is happening here, the meiosis 1 where it has stopped, the process is continuing by converting into secondary oocyte and also polar body. And this secondary oocyte is also again stuck at metaphase. It is stuck at metaphase. Okay, so it is not at meiosis 2. So in meiosis 1 we have said it is stuck at prophase. So in meiosis 2 it is stuck at metaphase. So when this will be released? It will be released after fertilization. So if fertilization will take place, then this secondary oocyte will convert into female pronucleus and one more polar body. So 2 polar bodies will be forming after fertilization. So what process is called ovulation actually? You say ovulation, right? Ovulation is the process where the primary oocyte is converted into secondary. This is the ovulation process. Okay, but in the case of fertilization, what happens is the secondary oocyte converts into the female pronucleus. Okay, so this is the difference which we need to remember. So actually this primary follicle, as I said here, it is stuck in the meiosis 1. Around it there are some follicular cells which come and they form a primordial follicle. And this primordial follicle only, whenever it is by LH surge, it will convert into secondary oocyte and polar body. So now let us see like how many follicles are present from the birth. So especially if you see in intrauterine life at 5th month, there are like about many follicles, 7 million follicles at the 5th month. And at the time of birth, there are 1 to 2 million follicles. So follicles are decreasing. First in intrauterine life at 5th month, 7 million follicles. At birth, 1 to 2 million follicles. By the time of puberty, there are only 4 lakh follicles. So you can see from the millions it has come to lakhs. And among this also only 400 to 500 follicles will be matured in entire lifetime of the woman. Okay, entire lifetime only 400 to 500 will mature, and that also 1000 will get atresia, will undergo atresia. And remember this initial requirement of all these follicles is not hormone dependent, there are hormone independent. Okay, so this is very important. Ovulation, is conversion of primary oocyte to secondary oocyte. Fertilization is conversion of secondary oocyte to female pronucleus. In intrauterine life, at 5th month there are 7 million follicles. At the time of birth, 1 to 2 million follicles. At puberty, 4 lakh follicles. And when in whole lifetime of a female, total only 400 to 500 follicles will get matured, and 1000 follicles will undergo atresia process. And all this process of follicles is hormone independent. Now coming to the structure of follicles, so if you see the follicles, especially the primary follicle, I mean like this primary oocyte. So around the primary oocyte there are corona radiata cell. This is corona radiata, okay. And here you have cumulus oophorus, okay. And totally here you have the granulosa cell, and here you have a antrum, and covering them you have internal theca layer, external theca layer, okay. So this is the structure of primary follicle, primary oocyte. So inside primary oocyte, around it corona radiata, below cumulus oophorus, around granulosa layer, antrum, internal theca, external theca. So now let us draw the secondary oocyte diagram. So here will be the secondary oocyte, around it will be the ooplasm. Around the ooplasm you will have the perivitelline space. This one is perivitelline space. So this is the perivitelline membrane. So outside the vitelline membrane you have zona pellucida, okay. So outside the zona pellucida you have the corona radiata like this. So corona radiata has come to us outside, if you see in the case of secondary follicle. So secondary oocyte. So when the secondary oocyte will form? During ovulation, right? This secondary oocyte will convert into pronucleus in the case of fertilization. So conversion of this primary oocyte to secondary oocyte is nothing but ovulation process. So this is the nucleus, this is the ooplasm, this is the perivitelline space, this is the vitelline membrane, this is the zona pellucida, outer layer, this is the corona radiata, okay. And of course one more thing, here will be the polar body, because there is a secondary oocyte, always there will be polar body. If you see two polar bodies, then you can say there is a fertilization, because after fertilization, female pronucleus and two polar bodies will be forming, okay. So we understood about ovulation, everything. And one more thing, as we said here, in the meiosis 1 prophase 1 diplotin stage, there is blockage of this primary oocyte conversion into secondary oocyte, that is ovulation process. So this process here, it is called as, this stage is called as Dictyat stage. And this Dictyat stage is not seen in spermatogenesis, right? Only in oogenesis you can see this Dictyat stage. And mainly, as we said, this meiosis 1 is continued because of LH surge only. And about polar bodies also we have discussed, and size. So coming to the size of ovum, the size of ovum is like about 120 to 130 micron. It is the largest cell in the body. So actually, if you remember, in the case of male, the length is of about 50 to 60, right? 55 to 60 microns. But female, you see 120 to 130 micron, largest cell in the body. And the size of follicle after ovulation will be like 18 to 20 millimeter. Just before ovulation, the size of follicle will be this much, 18 to 20 millimeter. So let us see, once again let us revise the, as we said, first the germ cell will reach the yolk sac at 3 weeks, it will reach the genital ridge at 6 weeks, it will reach the next oogonia formation will be at 9th week, next primary oocyte will be formed from 12 to 16 weeks, next follicle formation begins from 15 to 20 weeks, and follicle formation completed by 22 to 24 weeks. So all this we need to remember. So 3 week in the yolk sac, 6th week in the gonadal ridge, 9th week the oocyte, that is oogonia, is formed. Primary oocyte is formed 12 to 16 weeks. Secondary oocyte is formed, follicle formation layers, all those are 15 to 20 weeks. Finally, follicle formation is completed by 22 to 24 weeks. So 24 is the last. You can remember like the 3 table: 3 ones are, 3 twos are, 3 threes are, 3 fours are, 3 fives are, 3 six are. Okay. So remember the 3 table in order. So finally, let us discuss the events before the fertilization. Actually, the male sperm will reach the cervix of the female. So by reaching the cervix only, they'll start the process of capacitation, which will take like about 2 to 6 hours. Okay, and in this capacitation process, mainly this process is going on in the cervix. It has started in cervix, but the majority will be taking place in the fallopian tube, and that is the hypermotility of the sperm is getting in the fallopian tube but not in the cervix, but the process has started in the cervix itself. Okay, and next what happens is there is a process where all this will try to swim around the fallopian tube, like about only 300 to 500 will reach the fallopian tube, and for this process it will take about 30 minutes. So the like millions are coming, but only 300 to 500 are only able to reach the fallopian tubes within 30 minutes, and this now they will attach to the corona radiata, as we have discussed here about the secondary oocyte. Around there is a first one is a corona radiata, so they need to go to the corona radiata and release a hyaluronase enzyme. This is called as acrosomal reaction, so that it will reach the zona pellucida and attach especially to the receptor called ZB3 receptor. So only one will attach to this ZB3 receptor, so this will lead to entry into the perivitelline space. After it enters into the perivitelline space, actually the head, tail, neck, everything enters into perivitelline space. Here immediately now what happens is the secondary oocyte will complete its second meiotic division and forms a female pronucleus inside, and also the cortical cells here will form a cortical reaction, and also there is a zona reaction which prevents the other sperms to enter into this cell, so that only single one will go and fertilize, and it will lead to fertilization process. So if you see the order of these reactions, let me write down. The first one is capacitation. As I said, it takes about 2 to 6 hours. Mainly it starts in the cervix, but in fallopian tube it attains hypermotility. After that you have acrosomal reaction. So what is acrosomal reaction? It will release a hyaluronase enzyme and dissolve this corona radiata and try to enter into the cell. So after acrosomal reaction there is cortical reaction. Cortical reaction is where as the sperm enters this cell, the cortical enzymes are released. There is a cortical reaction. Finally, zona reaction. It is to prevent the polyspermy. So what is zona reaction? Only one can enter inside, so that the zona pellucida layer, after the entry of one sperm inside the cell, it will become more thick, so that other cells can't enter by means of this ZB3 receptors, so that the zona cell will become more thick. So this is the order: capacitation, acrosomal, cortical, zona. You can remember CACZ.